let's move on to today's session. Let me introduce today's speaker to you. So we are very happy to have Dr. Mahesh Kulkarni, who is a Senior Principal Scientist and Chair of the Biochemical Sciences Division at the CSIR-National Chemical Laboratory, Pune. He holds a PhD in crop physiology and has been a key researcher at NCL since 2004. His work centers on mass spectrometry-based proteomics and metabolomics with a strong focus on protein glycation in diabetes, age rate signaling, and PTMs. These studies have advanced the understanding of disease mechanisms and biomarker discovery. He has published over 130 research papers and was honored with the Chelaram Diabetes Institute Special Recognition Award in 2018. So Dr. Kulkarni plays an active role in the scientific community, organizing high-profile conferences such as the Proteomics Society's annual meeting and events on integrated omics approaches. So with this brief introduction, I welcome Dr. Kulkarni for his today's talk.
Thanks, Rita.
Yeah.
Can you hear me? Yeah.
Yes, now you're clear. So
Okay.
before I hand over to you, maybe those who are interested in our services, please scan this QR code and we can share more information after the talk. I'll just stop sharing, sir, and then you can share your screen.
All right. Yeah.
Yeah, please go ahead.
Okay. So now, do you see one slide or even the next slide?
I see one slide, but can you go full screen?
It's a full screen only.
Somehow it's not full screen for me. If anyone else can confirm.
No, we are also able to see all the slides on the left-hand side.
Yeah.
Huh?
There only one.
Okay. So, what do I do?
We'll change the.
Change the setting.
You go here, but put this in slide 0. Now.
Okay. Just give me a couple of minutes. Okay.
Yeah.
One minute. That one. Okay. How about now?
No, actually.
No?
Somehow I'm able to see it small screen. So you can make it full screen first and then select share screen and select that particular full screen, the slideshow one.
Okay, right? Yeah. So first make full screen. Yeah. Then go here. Share.
Share the entire screen.
Second screen. How about now?
Yes. It is full screen.
Yes. Now it's full screen.
All right. Yeah.
Sure.
Okay. So at the outset, first I would like to thank Venture Center team, especially Edna, Smita, and Rutuja, for inviting me to deliver this talk. So what I'll do is in next 30 to 40 minutes, I'll try to give a brief or brief overview of targeted mass spectrometry focusing on proteins and metabolites. And what I'll do is I'll give few examples of my own research. Okay.
50 mg के हिसाब से 50 mg में 3 माइक्रोमीटर तो जो भी तुम्हारा कॉन्सेंट्रेशन DTT
Requesting all the participants to kindly stay on mute.
So I would always like to begin with this. Yes. Okay. Not moving. I think the technical glitch. Now, can you see the full slide?
No, actually, it's
Not. How about now? No.
no, not yet. Earlier, when you had shared, that time it was.
No, but then the slides were not moving. Okay.
Maybe then in that case, you could try with the mouse first. Sometimes, with the downward key, it does not.
You need to click in this PowerPoint.
Or if you have a PDF, if you want to just convert it as PDF and then share.
Why so many?
This is a PowerPoint.
Is it? But that did not move. No? It's not moving, right?
Now try maybe with the mouse enter or the downward key.
No. Yeah.
Yeah. Now it's
Okay. Yeah. Let's get started. Okay.
yes.
So yeah, I always like to start with this WhatsApp can. So here what they have done is letters A to Z have letters A to Z were assigned values 1 to 26. Like for example, A1, B2, C3, and so on till Z as 26. And then what they do is they determine the value of different qualities like hard work, knowledge, attitude, and we know that attitude comes 100. Okay, so let's look this from a mass spec perspective. Now, if these A, B, C, D are amino acids and 1, 2, 3, 4 are the molecular weights or molecular masses, and let's consider this hard work, knowledge, luck, attitude as small peptides or proteins, and then we can determine their molecule. Like if we know their molecular weights, then we can determine what they are. Like we can determine the identity of these peptides. Like we can say that this is hard work, attitude, or these different kinds of peptides. So there the mass spec comes into picture. Like mass spec is the tool which helps in determining the molecular masses or of different compounds, peptides, proteins, and so on. So let's try to understand what is mass spec. Mass spec is an analytical device that determines the molecular mass of chemical compounds by ionizing and separating molecular ions according to their mass charge ratio. Okay. So one of the key requirements to determine the molecular weight is these molecules need to be ionized. Ionization occurs by either gaining proton or losing proton. Okay, so once the ions are formed, they are pushed into the mass spec and then they are separated according to their mass to charge ratio. The x-axis represents the M by Z mass to charge ratio, and the y-axis represents the relative abundance of this molecule. The mass spec consists of mainly three components: an ion source, as the detector. And of course, we require a sample inlet system. This sample inlet system can be a direct infusion, or you can hook to the liquid HPLC. These HPLCs, again, there are different kinds, like you can use UHPLC, nano LC, micro LC, and so on. Or you can use capillary electrophoresis and direct infusion. And in the ion source, we have electron ionization, chemical ionization. But in LCMS, we always use electron spray ionization, which is considered as a soft ionization and is highly suited for biomolecules like peptides, proteins, metabolites, and so on. Okay. And then once the ionization occurs, the ions move into a region of mass spec called mass analyzer. This is then very important because here the ions get separated according to their mass charge ratio. And we have again different kinds of mass analyzers: quadrupole, ion trap, time of flight, magnetic sector, and so on. And then we have detector. Normally, the new generation mass spec, they have electron multiplier as a detector. So coming to there are basically two kinds of mass spec: high resolution and high sensitive. So the orbit trap and time of flight mass specs are considered as high resolution mass spec. They will provide you a very accurate data with a resolution as high as orbit traps can do, starting from 17,000 to as high as 5 to 6 lakh. Okay. Whereas the top, again, the resolution of the top is about 30,000 to as high as 1 lakh resolution. But these are not as sensitive as triple quadruple machines. Okay, but they provide very accurate molecular weights. Okay, whereas triple quad is not a high resolution mass spec, but it's a high sensitive mass spec, and this is suitable for all quantitative applications. So conventionally, proteins, metabolites are quantified by approaches like Western blot, ELISA, immunohistochemistry, immunofluorescence, HPLC, and so on. But it's possible to quantify these metabolites, proteins, and peptides using mass spec. So there are mainly three different approaches. One is label-free quantitative approaches, um, such as DIA, data independent acquisition analysis. That path refers to sequential window acquisition of all theoretical masses, and MSc in waters. So these are all label-free approaches. And in labeling approaches, they have iTrack, ICAT, TMT, and all where you SILAC, where you need to label the peptides and then to relate to quantification. And in targeted mass spectrometric approaches, we have parallel reaction monitoring, and then we have MRM, multiple reaction. This, especially the MRM, is considered as a gold standard for mass spectrometry based quantification, and you can do absolute quantification using MRM approach. So targeted quantification is akin to quantifying a basket of different fruits, which is untargeted, and the targeted is quantifying one fruit at a time. Okay. So let's try to understand more about targeted quantification. So as I said, there are mainly two approaches: multiple reaction monitoring and parallel reaction monitoring. Multiple reaction monitoring is also called selected reaction monitoring. Okay. So we can either use MRM or SRM, and this is mainly this is done on triple quadruple mass spec. Okay, and quadruple act as an ion filters. You can select a specific precursor and fragment ions. Okay, the precursor ions are selected on using quadruple one, and they are fragmented on in the collision chamber, which is Q2, quadruple two, one or several of the fragment ions. Usually, 2 to 3 fragment ions are selected, and they are used for quantification. Okay, so let's explain this. So if you have a multiple analytes in a mixture of in your sample, multiple molecules are there. One particular ion is selected. In this case, this red colored ion is selected, and that is fragmented in a collision chamber, and you get several fragments. But among several fragments, you are now choosing the most consistent and abundant fragment ions. So here, this green, violet, and red colored fragment ions are selected. This selection is possible only on a quadruple based machines. Okay, on using triple quadruple. Whereas parallel reaction monitoring is also a quantitative approach, but it is not as sensitive as multiple reaction monitoring, because here the third quadruple is replaced by orbit trap or time of flight. Okay, so this generates high resolution full scan MSMS, and all transitions can be used to confirm peptide. Here, transition refers to in targeted mass spectrometry, transition refers to a pair of precursor and fragment ions. Here, you need not have to choose the fragment ions. Post acquisition, you can select one or multiple fragment ions, and that can be used for quantification. Why targeted mass spectrometry? Currently, most of the validation assay relies on antibody-based approaches like ELISA. In the absence of commercially available antibodies, MRM or PRM will be very useful. And MRM is considered as gold standard for mass spectrometry-based quantification. You can do absolute quantitation by MRM and even by PRM. Yeah. More than one protein can be quantified simultaneously. So you can quantify as high as about 100 proteins in about 30 to 40 minutes of acquisition time. Okay, so that's the advantage, and it's relatively less expensive compared to antibody based approach if you are quantifying more than one or two analytes. When do you use targeted mass spec versus untargeted mass spec? Untargeted mass spectrometry is done to identify maximum number of proteins or metabolite in a limited number of conditions. Let's say you have two samples, control and diseased, 2 or 3, and if you want to do discovery experiments and identify differentially expressed metabolites or proteins, then the untargeted is best suited. Okay. Whereas targeted mass spectrometry is useful for basically validation purpose. You want to identify and quantify limited number of molecules. Okay, these molecules could be proteins or metabolites in a maximum number of conditions. So in for example, in the discovery experiment, you have identified certain markers and you want to validate whether these markers are upregulated or downregulated in all the samples, like in hundreds of samples. There, the targeted mass spec comes. This is the workflow of MRM and PRM. So in both the cases, you need to select the target, and once the target is selected, you need to select the peptides, especially when it comes to protein. And once the peptides are selected, you need to select the transitions, which is fragment ions. So MRM requires selection of transitions, whereas the PRM doesn't require selection of transitions because the orbit wrap and time of flight don't have the ability to select the fragment ions. So once its transitions are selected, they need to be validated. Okay. So the next step is validation. And eventually we have to develop a calibration curve to quantify the molecules, that's proteins or metabolites. So selection of target protein. Okay. This target protein can be anything, like it could be an important protein in metabolic pathway, it could be a diagnostic marker for a particular disease, could be a post-translationally modified protein such as phosphorylated protein, protein complex. It totally depends upon what you are interested in and what is your research. Okay. So in my case, the interested protein or the what is my interest is glycation. So glycation, so what I'll do is I'll combine my research as well as the workflows and try to explain how these techniques can be used. So the my protein or my interest is glycation. Glycation is a non-enzymatic reaction between glucose and protein, and it is predominantly seen in diabetic condition. Because of the elevated blood glucose, the proteins undergo modification by glucose, and that is called glycation. So one of the most widely known glycated protein is HbA1c, okay, which is a uh, marker, for diabetes. So it provides the glycemic status of preceding 3 months. Okay. So the HbA1c is less than 8.7 in healthy individuals, and 5.7 to 6.4 is considered as pre-diabetes, and above 6.5 is considered as diabetic. So although this is considered as a gold standard, there are certain limitations in the conditions like anemia, blood loss, pregnancy, glycated hemoglobin or HbA1c is not an ideal marker. On the other hand, and it also doesn't predict diabetic complications. Like there are many literature to show that it doesn't predict complications like such as cardiometabolic risk and so on. While glycated albumin is emerging as an alternative marker because this is more closely associated with coronary artery disease. Okay, and it's an optimal marker for gestational diabetes, and it identifies pre-diabetes not detected by hemoglobin. So therefore, we try to understand the chemistry of albumin versus hemoglobin glycation. Albumin is the most abundant protein in the serum, and it has more number of lysine and arginine residues as compared to hemoglobin. It has 59 lysines and 26 arginine residue, whereas hemoglobin is a relatively small protein and has only 17 lysine residues. And albumin is readily accessible for glycation in the serum because it's really it is available for glucose for modification. Whereas hemoglobin, if it has to undergo modification, glucose has to enter into the RBC through GLUT1 transporter, and then the hemoglobin gets modified. And apart from that, glycated albumin also interacts with a receptor called RAGE and accelerates the progression of diabetic complications. So therefore, we have chosen glycated albumin as one of the important target protein. Apart from that, I'll be also giving examples of quantification of glycated insulin and glycated urea that I'll discuss once it comes. Okay. So now my target protein is glycated albumin. The next question is how to select peptides. Okay. Selection of peptides is an important criteria. Is an important. Okay. So a few representative peptides will be used to quantify each protein. So at least 2 or 3 peptides should be used to quantify the target protein, and it has to follow certain criteria. Okay. So it should be a unique sequence. Okay. And the peptide should be consistently absorbed in LCMS methods. And the peptide length should be about 8 to 25 amino acids. It should have good ionization efficiency. And M by Z should be within the range of your mass spec. And it should not be too hydrophilic or hydrophobic. Okay. If it's too hydrophilic, then it elutes faster. And if it's very hydrophobic, then it sticks to the column and doesn't get eluted properly. So when it comes to glycated protein, glycated albumin is a post-translationally modified albumin. So we didn't know which peptides we have to select. Okay. The routine way of peptide identification is we take the protein of our interest, digest with the enzyme protease such as trypsin. Then the peptides obtained are separated by LCMS, and the raw data is subjected to database search, and we identify proteins and peptides. This is the routine work. But in case of post-translationally modified proteins, will not be knowing the exact, especially if you are doing for the first time, you will not be knowing exactly which are the sites that are modified. So there comes the mass spectral library. Okay. So this is an important tool. A mass spectral library contains a collection of reference mass spectra comprising of precursor and fragment masses for various chemical compounds or standard peptides. These spectra are obtained through mass spectrometry experiments for standard compounds or peptides. Identification can be done by comparing an experimental mass spectrum with those in the ion type. So, for example, you have to establish the mass spectral library and then compare your sample with the mass spectral library. So, for this, what we have done is, so this is the redundant one. So, for this, what we have done is, we have developed a mass spectral library for selection of glycated peptides. So we modified the albumin with either glucose, glycolic acid, or methyl glycol. So the glucose modified albumin provided ammonary modified lysine, which is one of the glycation modification. Glyoxylic acid provided carboxymethyl lysine, and methyl glycol provided carboxyethyl lysine. Okay. So these differently modified albumin were digested with trypsin, and then they were acquired on LCHRMS, that is Thermo Q-Exactive Orbitrap, and then we perform the database search. So when we do the database search, so the database, the algorithms like Sequest or Mascot, they have the ability to identify PTMs, but sometimes they also provide false positives. So you have to manually inspect whether they are correct PTMs or not. And what we did is in this case, the proteome discoverer identified glycated peptides were manually inspected for the modification. So the way we have done is there should be an accurate mass shift in precursor ion corresponding to modification. So if this is a control peptide, okay, whose mass is 1128, so the modified peptide shows an increase in mass by 58 daltons, corresponding to carboxymethyl lysine. Okay. And this increase is less than 1 ppm. Okay. This is at the precursor level. And then we perform the MSMS. So those who are new to the peptide fragmentation, so when we perform peptide fragmentation by CID, we get two types of ions, B ions and Y ions. From amino terminus, they are called B ions. From amino terminus to the first peptide bond, it is B1. Amino terminus to second peptide B2, and so on. Similarly, in the reverse order, carboxy terminus to the last peptide bond, it's called Y1, Y2, and so on. So typically, like let's say if we have this peptide, and if this is fragmented, this leads to generation of B and Y ions. The B will be K, KQ from N terminus, KQ, KQT, and so on. And from carboxy terminus, it's in the reverse order, like K, BK, LBK, and so on. So this is a typical pattern of fragments you get for an unmodified peptide. But let's say the first lysine is modified. The lysine here, this is modified with carboxymethyl lysine. Okay. So if this is modified, your B1 will be a lysine mass plus modification mass. So in this case, 58 is added. Since this lysine is present in all the B ions, so all the B ions now show an increase in mass by 58 atoms. Okay. Whereas the Y ions remain same between unmodified and modified peptide. So this I'll show you here. So this is an unmodified peptide, okay, whereas this is a modified peptide. So these are the fragment ions, and here this is a zoomed version. Here the B1 is 129, whereas this B1 is 187, which is 121 plus 58 becomes 187. So likewise, B2 is also shows an increase in B2, and B3 also show an increase in mass by 58 atoms. Okay. So this way we characterize 50 glycated peptides corresponding to 23 lysines. And then we constructed the mass spectral library, which consisted of precursor mass, charge state, the retention time, and all the important, the most intense fragment ions. This information is very important, especially if you want to quantify these peptides by MRM or PRM. Okay. So further, what we did is we identified since there were 50 glycated peptides and there were 23 lysine residues. So to cut down the number, we identified 3 lysine residues, that is K36, K438, and K549, to be highly sensitive for glucose modification or glycation modification. And we term them as glycation sensitive peptides. And these peptides we quantified by an approach called parallel reaction monitoring. Okay. So the parallel reaction monitoring is done on Orbitrap machine. So what we did is we quantified these 3 peptides by this approach in 20 healthy and 20 pre-diabetic subjects, and then the quantification is done by an open source tool called Skyline. Okay, so what Skyline does is it generates the chromatogram of co-eluting fragment ions. Okay. So you are seeing that there are different colored peaks. Okay. The purple, brown, orange, and so on. So these are co-eluting fragment ions of modified peptide. Like so, and we consider these areas and then compare between healthy and pre-diabetic subjects. So as expected, so the abundance of this peptide was higher in pre-diabetic condition. Whereas this is another peptide, SCHCIA, which is an unligated peptide. And the abundance of this did not differ much between healthy and pre-diabetics. Okay. So when we did the fold change analysis of these peptides, so these three peptides showed a higher fold change, more than 1.8 fold change, as compared to HbA1c, which showed only 1.16 fold. Okay, suggesting that these peptides determine prediabetes better than HbA1c. Okay, so this is how we quantified glycated albumin peptides using parallel reaction monitoring. Next comes the multiple reaction monitoring. So in multiple reaction monitoring, as I said, this is done only on a triple cord mass spec. So here, one or multiple precursor ions are selected one at a time and fragmented, and the fragments, uh, like 2 or 3 fragments, are selected for quantitation. So therefore, now here the question comes is which transitions or which fragment ions we have to select. So for this, what we have done is we have quantified glycated insulin by MRMR approach, but the selection of transitions can be done by software such as again Skyline, which predicts the important transition ion, like it ranks. For example, you see here, this is a peptide, so it provides the like there are different 1, 2, 3. So 589 is the like when you do the MS analysis, you will get this to be the most intense in with highest intensity, followed by 490 and 361. So it also predicts. It helps in selecting the transitions. Okay. Okay. So coming to the MRM based quantification, here we used insulin. Here our interest was to quantify glycated insulin. Like albumin and hemoglobin, insulin also undergoes modification by glucose. This arginine 22 and lysine 29 of the B chain of insulin gets modified by glucose, and previous studies have shown that glycated insulin has a reduced biological activity. So therefore, we wanted to quantify, and we have also shown that glycated insulin uh, causes insulin resistance and activates age-range axis. So to quantify insulin and glycated insulin, what we did is we synthesized uh, glycated insulin and performed the ESIMS. This is a MALDI MS, whereas on electron ionization, you get the multiply charged ion. For example, in MALDI, you get a single peak of 5808. But the same insulin now here shows different charge states, starting from 968, 1162, 1452, 1936. So I have given you here an example. To multiply 968, which is a 6 charge state, into 6 and subtract 6. So the mass will be 5807. Okay. You can do this for all other charge state. And then we developed the transitions, okay, of different charge state, like we took the standard human insulin and we selected a pair of precursor and fragment ions like for insulin, methyl glycol modified insulin, glucose modified insulin, and D30 is a mutated insulin, which is used as a standard. So this is how a typical chromatogram looks. So this is control and this is diabetes. So this is insulin, glucose modified insulin, and methyl glycol. The next thing is we have to quantify. For quantification purpose, this is the formula is used. Peak area ratio of your analyte, which is the analyte peak area by a standard isotope peak area. Okay. Then analyte concentration equals to peak area ratio into the concentration of stable isotope peptide standard. Okay. So to develop a standard curve, what we did is we took insulin in different concentrations, okay, and then developed a calibration curve performing MR-MRCs. And then we quantified insulin, glucose modified insulin, and methyl glycol modified in healthy pre-diabetic and diabetic subjects. So if you see here, in all subjects, healthy, pre-diabetes, and diabetes, in all the subjects, we have detected and quantified glucose as well as methyl glycol modified insulin, suggesting that even in the healthy conditions, there is a lot of formation of glycated insulin. Okay, that could be one of the trigger for developing insulin resistance and eventually diabetes. Okay, so whenever there is a hyperglycemia or insulin resistance, there is a lot of load on beta cells to produce more and more insulin. But because of hyperglycemia, that gets glycated, and this glycated insulin has a lesser activity, lesser binding activity towards insulin receptor, and it binds more to the RAGE, which causes a generation of reactive oxygen species and inflammation. So this in turn again causes insulin resistance and hyperglycemia. So therefore, it's a kind of a vicious cycle, and it's important to quantify and monitor glycated insulin. Coming to the another topic, metabolomics, quantification of metabolites by targeted approach. So in this case, we have addressed diabetic nephropathy. Okay. So here, what we have done is we looked for a glycated metabolites. Okay. Currently, diabetic nephropathy is detected by microalbumin, okay, and albumin creatinine ratio. But other markers of kidney dysfunction are accumulation of creatinine in the serum and accumulation of urea in the serum. Okay. So the way urea produced is whenever the protein is metabolized, okay, so it leads to formation of urea. And urea has the two amide groups, NH2 and NH2. And this can easily get modified by glucose, okay, to form monoglycated urea and diglycated urea. Okay. So, what we hypothesized is, so whenever there is a kidney dysfunction, urea starts accumulating in the serum. However, in diabetic conditions, if the blood glucose levels are high, it gets modified by glucose and forms monoglycated urea and diglycated urea. And therefore, probably, although the urea is accumulating much earlier, but it is getting modified and maybe not getting detected, and we are somehow underestimating the urea formation. So to address this, what we did is okay. So, this is the routine metabolomics workflow. The way metabolomic analysis is done is untargeted metabolomics, where you extract the metabolites from serum and do mass spec and use software to identify the metabolites. But since this urea is a modified glycated urea is a modified amide, so we do not find in this, you cannot detect this using an untargeted metabolomics workflow. So for that, you need to do a targeted metabolomics. So what we did is, again, here mass spectral library becomes very important. So what we did is we took several amines, including amino acids, creatine, urea, and we modified it by the glucose, and we synthesized different glycated amino acids and glycated urea, and generated the mass spectral library for that. So using that information, then we quantified these metabolites in the serum of diabetic and healthy subjects by parallel reaction monitoring. So I'll show those details. So this is the glycated urea. Okay. So urea, the molecular mass of urea is 60, and when it reacts with the glucose, it's 160 gets added, and here 223 you see. Okay. And similarly, 385 is the diglycated. Okay. So in this approach, um, in the whole workflow, so once you select the transitions, you have to validate the transition. Okay. So to validate the transitions here, what we did is we synthesized glycated urea using both 12C glucose as well as 13C glucose. Okay. So the 12C glucose showed 223. Okay. Whereas if we use 13C glucose, here also 13C. So there was an increase in mass by 6 times. Okay. Same way, when it was fragmented, so this is a 104 is one of the fragment. This fragment showed an increase in mass by 2 carbons, 2 daltons, 106. So 116 showed 120. So suggesting that these are indeed the true fragments, because when we use 13C glucose, the molecular mass of these fragments is increasing. So this is how validation is done. Similarly, for diglycated urea, so it is a 385. So there will be addition of 12 Dalton. 385 plus 12 is 397. Okay. And then the same fragment, 203, now it shows 209. So here we are validating the transition. And then these transitions were used for quantification. So we quantified both monoglycated urea and diglycated urea in healthy diabetic subjects with no complication and in diabetic nephropathy condition. So what we found was in both diabetes and diabetic nephropathy condition, the both monoglycated and diglycated urea levels increased, and they showed good correlation with the fasting blood glucose and HbA1c. Given that urea gets converted to glycated urea in the hyperglycemic condition, it is crucial to quantify monoglycated urea and diglycated urea along with the urea for the diagnosis of diabetic nephropathy. So now this study we are extending to large number of samples, and let's see how it goes. So with this, I thank all my students, collaborators, CSR, Director HCL, and all the previous heads of the Biochemical Sciences Division. So I'll be happy to take questions. Okay.
Thank you, Dr. Kulkarni, for covering the topic so nicely and explaining us the targeted mass spectrometry. So now the session is open for question and answers. I would request participants to kindly post their questions in the chat box, or maybe I request our admin team to kindly allow you to speak so you all can unmute and ask your questions. So I think Rishabh has asked in the chat box if we can get these slides.
Okay.
So,
So what is the question?
he wants to get these slides. So maybe you can only answer if the slides some
Yeah,
common things.
share the slide. Yeah.
Okay, sure.
Yeah.
So you can share it with us and we'll share it with the participants who have attended.
Sure. No other questions?
Any questions from any participants? You all can unmute and ask. So there are no questions in the chat box.
Okay.
There were just a few comments and requests.
All right then. Yeah. If there are no questions, then
Yeah, if there are no questions, I've just posted the PC Analytical Services LinkedIn handle. You all can please connect with us and just give me a minute. I'll just paste the feedback link also. Your feedback is very valuable. Kindly fill the feedback link and let us know how did you find this talk. So Dr. Kulkarni, I think there are no questions. Yeah, Dr. Uma has one question. Dr. Uma, you can please unmute yourself and ask the question, or you can post it here. Oh, you are unable to unmute. Oh, wait, I'll just maybe you can type it in the
yeah,
chat box.
type this.
I'll just try. Dr. Uma, you can try now if you are able to unmute. Actually, I've asked our admin team, they had changed the settings, but after the talk.
Hello, ma'am. Hello.
Yeah, now we can hear you. Please go ahead.
Okay, ma'am. Thank you, sir. Hello.
Yeah,
Hello.
yeah, I can hear you.
Sir, thank you, sir. Thank you for your very wonderful session. I have one question, sir. We are fermenting black soldier fly biomass with lactic acid bacteria, sir. After fermentation, we want to know what are the changes in proteins and what are the metabolites that are going to be secreted in that biomass. So can we use this targeted mass spectroscopy for identification of those metabolites?
So the targeted mass spectrometry is used only when you know the metabolites already. Okay. So you need
Okay.
to actually first identify what are those metabolites. Okay. So once
Okay.
identification is established, identity is established, then you can use the targeted approach for quantification.
Okay, but identification we can unknown compounds we can't identify.
No, that can be identified by untargeted approach. Like you can use a QV exactive MS and then metabolomics and identify those metabolites. Once the identity
Q-TOF and all.
Q-TOF also. Yeah, you can use exact or QTOF. Once the identification is established, and in subsequently, if you want to quantify this in different batches or different samples, then
Okay.
the target comes.
Means only for quantification, targeted is useful.
Correct.
Okay. Okay, sir. Thank you. Thank you for your information.
Yeah.
Are there any other questions from participants? Kindly unmute and ask the question.
Hello, ma'am, can you hear me?
Yeah, we can hear you.
Hello, sir. Thank you for your very informative session. I have a question on behalf of my friend, actually. She had some plant extracts which showed anti-quorum sensing effect. Then she actually fractionated them and sent those fractions for analyzing what are the metabolites in them. So she got metabolites and they gave the data in both positive and negative mode. But there are like some hundreds of metabolites in positive mode as well as hundreds in negative mode. So suppose we are writing this information in a paper, how do we condense them? Because this is, these are
Yeah.
the, yeah.
So what you should do is, okay, you have identified 100 metabolites, okay,
Yeah.
in both positive, negative in different samples.
Yes.
I think it's also important to consider their areas, like which are more abundant, which are less abundant. Okay.
Yes.
And how they are related to your work.
Yes.
So only those you need to consider.
Okay, so because I think when she saw the past papers, review papers, like there are not many papers on what are the actually metabolites which have been found in those plant extracts which she has tried. Probably some related papers also we could check out then, no, sir?
Yeah, you can check. So what is the question she's addressing?
She is actually, it is just the metabolite analysis of those fractions. Those fractions have showed the antiquorum sensing effect.
So how do you say that they are anti-quorum sensing? Like,
so she has done some assays and everything, and she has come to that conclusion. She has extensively studied about those plant extracts, and then fractions also she has.
correct. Not all the metabolites will have this activity, right? Only a few of them may have, and like
Yeah.
film formation or anti-quorum sensing.
Yes, a
Correct.
few of them may have, or they might have to different degrees also. We don't know.
No, yeah. So I think, yeah, you have to maybe fractionate them and then study again anti-quorum sensing activity.
Okay. Okay, sir.
Okay.
Or we could relate to the previous literature also, as you said.
Yeah, right.
We can. Okay, sir. We'll try that. Thank you, sir.
Okay. Yeah.
Sorry, I'm so sorry. I was on mute. So I was thanking you, Dr. Kulkarni, for sparing your valuable time and sharing the insights with us. So again, request you to kindly share some of the slides. If there is something confidential, you can kindly take that out and we'll share them with the participants. I hope all the participants have filled the feedback form.
What I'll do is I'll share the workflows. Okay.
Okay. Sure, yeah. We'll share that with the participants.
Okay. May I, Yeah.
Yes, sir. Thank you very much. You can leave. Maybe I'll just keep the meeting on for another 5 minutes for people to take a look into the chat box and fill the feedback. Thank you again.
Yeah, I want to ask, like for example, sometimes our mass spec don't work. So, what are the charges of your facility, like for metabolomics, proteomics?
Sure. So it's there on the website, but separately we'll share it over email details.
Okay.
Yeah. Okay. I'll just ask our team to immediately share it with you.
Okay, thank you. Yeah, I'm leaving now.
Yeah. Thank you very much.